This is the third part of our discussion of the cerebral cortex. In part 3, we're going to focus on the occipital lobe. The occipital lobe is the location of the primary visual cortex and the secondary visual cortex and the start of the visual association area. The primary visual cortex is where we initially have visual perception. While here it recognizes things such as shape and movement and dark light, it does not really interpret those things. That gets interpreted in the secondary visual cortex, which is where the information is going to go to next. You'll recall from our previous unit that the visual information from the thalamus arrives at the primary visual cortex via either the superior or the inferior optic radiation. Bilateral lesions of the complete primary visual cortex leads to cortical blindness. But the individual may have blind sight because the information is still getting to subcortical structures such as the thalamus, the hypothalamus, and the superior colliculus. Unilateral damage of a primary visual cortex results in homonymous hemianopia. Again, there may be blind sight in this area of lost visual field because the information is still getting to those subcortical structures. On the lateral view, you can see the primary visual cortex here shown in the blue oval. I want you to note that the majority of the primary visual cortex cannot be seen on a lateral view. It can only be seen on a medial view where the calcerine fissure is located. That is where the bulk of the primary visual cortex is located. And here on the medial view, you can see the primary visual cortex on either side of that calcerine fissure. You might also recall from the last unit that if we were to damage just the superior bank of that fissure, we would have a quadratopia. So if it were in the left hemisphere, as this is shown here, we would have a right inferior homonymous quadratopia. If the damage were on the lower bank of the calcerine fissure and the upper bank were intact, we would have a right superior quadratopia. And of course, we would have left quadratopias if the injury were instead to the right hemisphere. The secondary visual cortex then surrounds the primary visual cortex and receives input from the primary visual cortex as well as information from the superior colliculus. This is where we interpret movement and color and various properties of visual stimuli. This is the area where there may be some altered function of some of the neurons, resulting in color blindness, which is more common in men than in women. Our secondary visual cortex sends this visual input to a lot of different places, as you can see here. So on the lateral view, you can see now in the light blue the location of the secondary visual cortex. So it's essentially the rest of the occipital lobe. And again, you can see it here on the medial side. So we have our very localized primary visual cortex predominantly on the medial aspect of the occipital lobe on either side of that calcerine fissure. And then it is completely surrounded, both medially and laterally, by the secondary visual cortex. And this is the remainder of the occipital lobe. If we have damage to the secondary visual cortex, we get what is called visual agnosia. And this is the inability to recognize objects by sight. This is not the only location we have visual agnosia. This is kind of the first location where we might see it. Generally, we're able to describe the size and the shape of the object because we can get that from our primary visual cortex, but we can't really identify the object by sight because the information that is necessary for that gets sent to another part of the brain in the temporal lobe. And if the information cannot get to the temporal lobe cannot interpret it. So this is not the area that actually determines object recognition, but it sends information to the part of the brain that does that. And as I mentioned, color blindness is associated with injury to this part of the brain, but is also associated with congenital absences of certain types of neurons or activation of certain types of neurons. And then injury here can result in motion blindness, where you don't recognize or understand the movement. You don't interpret movement of items in your visual field.